Hello, everybody. I'm Dr. Deanna Minich. Welcome to another one of our Thought Leader Conversation series. And these have been, I must say, it's been a privilege to be part of these and to be having these interviews with all of these different experts and learning from them. And I hope that you, if you've missed any of these, that you can go to the website, theana.org, and look at the archive. We've done a number of these already. So today's guest, I must say, I was reviewing Dr. Mason's bio, and I was thinking, oh my gosh, my eyes were as wide as saucers just thinking of all the things that he's done. Let me just give you a little bit of his bio, his background. He's currently an associate professor at Weill Cornell Medicine with appointments at the Tri-Institutional Program in Computational Biology and Medicine between Cornell, Memorial Sloan Kettering Cancer Center, Weill Cornell Medicine, the Sandra and Edward Mayer Cancer Center, and the Family Brain and Mind Research Institute. My goodness, these institutes. You're really like the point of synthesis for so many of them. He's also the director of the World's Quant Initiative for Quantitative Prediction, which bridges prediction methods in finance with genomics. We have a superstar, everybody. In fact, right before we went on live, Dr. Mason was just a hair late. He said he was just coming from the hospital. He was just collecting samples. So welcome, Dr. Mason. Very nice to have you today.
Yeah, it's a pleasure to be here. Thanks so much for a very kind introduction. I'm really happy to be here.
Well, I didn't even finish reading your whole bio. You're just amazing in terms of all the things that you're doing. You're a wizard bringing together many different cross disciplines. So why don't you tell us where you were just now and what you're in the process of doing?
Yeah, so we've actually been, as most people know, New York City was hit really hard with the COVID-19 pandemic, and we've been working directly with the hospital, multiple hospitals actually in the city, working on getting samples from patients as they come through to help with diagnostics, to make it faster for testing and seeing what strain of the virus is present, really helping with contact tracing and rapid diagnostics, and then also looking at what happens inside the bodies of people that are sick. So looking at everything from the gut microbiome and how that changes to their inflammation markers, blood work, and also looking, of course, at the site of infection, the nasal pharyngeal swabs. So we're looking at everything we can to learn as much as we can to attack the pandemic and hopefully avoid maybe a next one.
So what are you finding so far? Or are you in the mode right now of collection before you get to the analysis? Or are you analyzing real time?
It's all the above. We're basically constantly getting samples, and then what we do is to examine the samples. It's similar to what we do at other discussions we've had with the ANA, is looking at, say, microbiome profiling, or looking at all the bacteria, what's in your gut, or how do you think about the sort of mini pharmacy inside your body. One of the things we do is called shotgun RNA sequencing, which means you basically, kind of like the shotgun, breaks apart all the cells, ruptures all the fragments of RNA, the piece of nucleic acid inside the cells, and then you just characterize all of them, you sequence them all. We're looking at this from patients, and we can see the virus is present, what it's doing to the cells. And we just actually put online the first 735 patient samples, their data, what we see for the results. It's really interesting. We can see that, the virus really, of course, makes people sick and disrupts their immune system, disrupts most of their hematological network, so how they even process blood. And even some of their olfactory receptors are changing. So the way that they can smell, some of the phenotypes have been that people can't smell when they get sick, and they can't figure out why. We can actually see it in the molecular data that the genes that control smell seem to be perturbed. And so it's really interesting to see that kind of data.
Can you tell us what are the genes that control smell that you might be assessing? Maybe just a couple.
Yeah, well, there's actually, they're called olfactory receptors. They do most of the sensing in the nose and in the airways. They're the largest class of genes in the human genome. So if you think about how many genes are there in the human genome, there's tens of thousands. By the latest count, there's probably 60,000 total human genes, some that make proteins, some that don't, some that are old genes. Olfactory receptors are the biggest class. There's actually almost 2,000 of them, and it's because they have very exquisitely control which ones get turned on in your cells in your nose to let you smell and taste things. Actually,
how fascinating! Why do you think that is? Why would the human genome skew towards olfactory-related genes? Talk about that as it relates to biology, survival, or even now in modern day life and our physiology.
Yep. Well, what's interesting, there's a comparative evolutionary aspect to this. If you look in other primates, some that have wet noses or some that have dry noses. From evolution, it's called haplorhines or strepsirrhines. But the different primates, like if you think of a lemur or something, or like a gorilla, almost every other primate has more olfactory receptors than we do. So there's some idea that we've lost them over time because we don't use our nose as much, maybe as other primates do. So there's other species that have a lot more. That I thinking is that if you really needed to survive, and you think of like a dog that's always sniffing things, right? They have a lot more olfactory receptors. They use their nose a lot, and we use it okay, but not as much as we maybe could. Some of the genes are also called pseudogenes, meaning they're kind of non-functional anymore. They're just kind of hanging around, just like a evolutionary relic that's just in your DNA still.
Hmm. And do you see any combination or combined effects with taste receptors? And so usually when I think of smell, I think the connection with taste is right there. And so we know about the bitter taste receptors and how they're throughout the body. They're just not on the tongue. They are in the gut and in the brain. So what do you see as far as that relationship goes?
Well, at least in terms of COVID, we haven't yet looked at some of those data, but we would expect that if the virus is making its way around the body, like for example, it's been found in stool, but it hasn't been seen in urine yet. We've contrasted like a flavivirus like Zika, which has been found in urine. So every virus has different kinds of tropism, which just means it goes to different parts of the body in different ways. And that depends a lot on the receptors. And some of the audience has probably heard about ACE2 as a receptor. Like, how does the virus get into the cell? And every virus, usually has its own favorite entry point. For HIV, for example, making AIDS, it's CCR5. And so just which virus has its favorite entry point. It's just kind of the luck of the draw. But we've seen that in the body. I don't know what it would do to the other olfactory receptors or taste receptors. We're just starting to look at that now. That's a good question, because it's probably doing something that's disruptive based on what some of the patients have observed. The other really weird thing, I don't know if you've heard of the happy hypoxic people for COVID, where there's
Yeah.
people who have really low amount of blood oxygen, which normally would make you really tired and groggy and even ornery, but they're fine. They're checking email, happy, and talking with people, and the physicians will come by and be like, "Did you know you have really low blood oxygen? You should be like almost passing out." And the virus seems to do something to some certain patients where they are just fine with low oxygen, at least for a while. So a lot of mysteries we're still looking at as we speak.
What about nutrients? What about nutrient regulated genes, and even to the point of happy hypoxia? Do we know anything about the role of iron? Do we know anything about, I know I was just looking at an article today with selenium and seeing how different regions of China were faring differently with COVID-19 based on the selenium that was available in the food supply of the
Interesting.
different regions. So what can you tell us,
Interesting.
being that, we are tapped into thinking about nutrition as a first line defense in the way of the immune
Yeah.
system? Can you tell us
Yeah.
anything about what you're finding with nutrients?
We have seen in some of the urinalysis that the iron levels are fluctuating a lot, that some of the nutrient levels do seem to be really variable between patients. We actually we got a huge dump of all the hospital record data on Friday. So we've just started looking at some of these factors now. Like what can we see from the blood work and the metabolomic work? Like what is in the body and how are they processed differently? So we have seen some differences, but it's very, it's only a few days old. So I can't say yet what the reason is, because some of it could just be by chance that we see different levels of iron in the patients that seem to be COVID positive versus negative. And we're also matching that with data at another hospital to confirm it there. But yeah, the China, it could be micronutrients, it could be variations of macronutrients that are there. We've just started to look at that, but it's almost certainly playing a role.
Hmm. Yeah, I just wonder, it would be so interesting to start to go into dietary patterns, nutrient patterns, different SNPs, different
Yeah.
polymorphisms for how we process these, and even looking at ones relating to biotransformation, like glutathione has come up, which I'm assuming that that's why selenium is so much a factor, aside from all
Yeah.
of the many different roles of selenium as well.
Yeah, yeah. Well, and also, a lot of for a lot of people on medication, like the majority of adults in the U.S. are on at least one medication, and so we also started to look. One thing we've already seen on the records that is, a lot of these medications need cofactors depending on what you're eating and for them to fully function. And so we've also started to look at what are medicines people taking pharmacologically versus what are they seeing nutritionally as a difference, and merging those data. There's already some really interesting surprises, like a lot of people would take ACE inhibitors, which are just for treating hypertension as a class of drugs. They so far seem like they're actually much more likely to get infected, almost 2 or 3 times more likely to get infected versus a controls of people taking any other drug. but then they don't seem more likely to die based on our records. So it seems to be, in some cases, it
Hmm.
increases your risk of getting an infection, but doesn't necessarily make you a higher risk of dying. If anything, people on these class of drugs seem to have a slightly lower chance of dying, but a higher chance of getting infected. So it's
Huh.
the things that seem counterintuitive, but it's because of the complex, pharmacy inside our body that is probably affecting the virus in some way. Maybe it's easy to get in, but then it doesn't do as much damage once it gets there.
Fascinating. Are you seeing any other correlations with other antihypertensive agents and drugs? So whether it's diuretics or beta
Yeah, beta
blockers, you.
blockers. Yeah,
Mhm.
so not yeah, we actually did an orthogonal analysis to see, well, maybe beta blockers showed the same thing. We didn't see that, so it seems to be, so it's given us a lead, and this lead is only really a few days old. So we're just try and put it more and get more and more patient data to understand, yeah, how does it link to their gut microbiome, or, and some of the patients also went through, we talked like, is it something coming from a change in a species? Is it a different virus that's also infecting some parts of their body? So all those questions plus the other drugs we're just looking at now.
Just give us a high level overview of where we are with COVID-19. Here we are, it's May 4th. And so we've been in homestay for about 7 weeks, 7 or 8 weeks, depending on when the state that we live in went into that notice and sent
Yeah.
that word out. So where are we now? Like, based on all of your data that you've been collecting, are we flattening the curve? When do you see us coming out of this? Do you see any reprieve for us in the next couple of weeks or even in the next months?
Uh, I think we are going in the right direction. So the overall news is that I think in most places the worst is likely past, but in other places I think it's still coming. Wisconsin, for example, just had its worst day 2 days ago, and Georgia had one of its worst days 3 days ago. But it was also, now they're open. So I think it's going to be very state dependent, and everything we've seen from the epidemiology and the phenomenology of the virus in patients is that we'll probably see a second spike in a lot of cases. So I think it's less a question of when do we go back to pre-COVID state, and how do we. It's more a question of how do we go forward now post-COVID, which is, it's not, there's not going to be a return to normal. There'll be some time of social distancing for probably months, and there'll be a lot of eyes on the horizon for what happens in the fall. When anytime someone gets a cough starting September 1 onward, they're going to think, oh crap, do I have COVID? And they're going to want to get tested. And you think about hundreds of thousands of, students in dorm rooms at one campus alone, like again, in Wisconsin, uh, they're going to all, it could be a recipe for a big second wave in the fall. And that, for big pandemics we've seen, for the 1918 flu, that was the one that was often called the Spanish flu, although it really didn't start there. It's just they were the ones born in World War I, so they were actually reporting bad things. They the second spike was worse for them. So we really, that's why we've been working really long on hard hours, is to learn as much as we can as fast as we can, because it looks like it's going to hang around. But but I think in terms of the near term pandemic, it's getting flatter. It's slowly getting back to a space where we can start to go outside, start to see each other again.
So, based on what you've been finding out, how can we be preparing? So, if we were to look out the next couple of, let's just say, 3 to 6 months, and then we see a resurgence, what can each of us do in the way of maybe food, lifestyle, what you know about physical activity, even our microbiome? I mean, some of the findings that you've been uncovering, what could we do to be proactive? Because I think that, this is an opportunity. As much as it's a crisis, there's also an opportunity here to
Yeah.
really figure out how we can bolster our defenses. So what would you
Yes.
say?
Yeah, great question. I mean, it's going to be as whenever possible, like most things in medicine and health, is a really holistic view of everything. So from the genes and the molecules all the way to the food you eat, and of course exercise and sleep. Those are all really key factors for strengthening your immune system. And we just finished a clinical trial with Longevity, which is a company. As a full disclosure, I helped found it, but the trial was just to see, can we decrease symptoms for people that have it was pre-COVID, but the clinical trial just got published, which is to see, well, if you give probiotics, and how much can probiotics help, or even prebiotics, which help you digest food. Both of them seem to be helping IBS, IBD patients, and you could decrease symptoms. So I think it's going to be, more trials like that, so that we need people to start to share their data and share clinical trial data, and also get as much information on what people are taking. But I would guess, based on trial data like that we've just published last week, and other data sets from other groups, I'm thinking it's going to be, obviously as much of a good, whole food diet that you can maintain, but then most of the probiotics or prebiotics are often, they certainly don't do much harm, and they probably, at least in certain cases, might, keep symptoms at bay. Although, I can't say that for COVID because we just haven't done the trial, but and we know in other indications of where you can have GI stress, which is one of the symptoms of COVID. Actually, some of the people have, outside of the funky loss of taste thing, some people have really bad diarrhea or GI symptoms, and then also obviously the low blood oxygen. So in that case, that's a hypothesis, but I think it's reasonable, and that would probably be helpful.
So how has this changed your life? Have you done anything differently? I mean, it sounds like probably your activities have ramped up.
Yeah,
So
yeah.
what about your level of stress? Are you able to be at home? I mean, it sounds like you're back and forth at the hospital, at home. And so
Yeah,
what has your
it's
life
been back
been
and
like?
forth. It's been completely crazy. It's actually, I'd say, for the past 6 weeks, I think it's been, most days started about 6:30 or 7, and they ended about 10:30 or 11, and it's been like, and Saturday and Sunday haven't been different. So the hardest thing is just, my daughter's here. We're doing homeschooling, and so I'm here like all day, but I barely see her, and so same with my wife. So I feel that's the hardest thing, is just not getting as much time with family, even though I'm literally in the next room. It's just constantly either running back and forth to the hospital or on calls, or they're doing logistics. Um, yeah, so that's been the hard thing, but it's starting to get a little bit more sane. So I think, it was really an all hands on deck moment where I've really just never seen so much generosity from the clinical staff, from the research staff, everyone wanting to help anyone they could. Even donations. We got a donation to Cornell from Citadel, who just wanted to help, so like people were really doing anything they could to send supplies, send cash, send, any people to come and help as well. So it was really heartwarming in that regard. And also I did get some time to help my daughter. I taught her how to ride a bike. So that was actually pretty great. Yeah.
Yeah. And so tell us what it's like in the hospital setting, so you just described what it's like for you at home and then running back and forth to the hospital. What are hospitals like now? And so just maybe you can just talk about the precautions. What about the first responders? Just kind of give us that milieu for those of us who aren't entrenched like you are.
Yeah, it's really like, in New York City in particular, it was really, really haunting for a while. So I think, and again, it's not so bad now, but about 2.5 weeks ago, right at the peak, there was a time where you could hear sirens all day, every day. And not, you've heard some sirens or one or two sirens before. If there's a fire, you've probably heard that in your life. But, you could just stand up in your bedroom and you could hear 4 or 5 sirens simultaneously, and all just picking up COVID patients. And so that continued for about a week straight of just constant sirens, which was the sound of people being picked up. And then at the hospital, it was, the ICU units weren't built for this many people. So a lot of the other operating rooms actually get quickly converted to ICUs because elective surgeries were stopped anyway. And then also the Hospital for Special Surgery, which is across the street from New York Presbyterian, does mostly orthopedics and other surgeries, and they weren't doing them. So they converted most of their rooms to ICUs. And, we had the USS Comfort outside of New York City for a while, which had some patients there. So it was really, but really I want to commend like the whole hospital and the staff, because even though it was in the middle of the worst peak of the worst pandemic in all of our lives, it was, everyone did everything they could to help each other and work as fast as they could as teams. And PPE was low for a little while, but that got better because also other hospitals, if they had too much, they'd send it to us, or anyone who just wasn't as badly hit. So, but it was a lot of, just lots of logistics that had to be ironed out to make sure people had what they needed. But overall, not so bad, I guess. Looking back, at the time, it just seemed like there was never an end of the things to do, or the emails, or the calls, or just getting supplies laid up. But it's mostly been organized all right, I think. I'd say.
So when you go to the hospital and you're actually dealing with contaminated samples, right? With
Yeah.
samples from people with COVID-19. So what kinds of precautions are you taking? Like, how do you garb up? Just kind of walk us through all of that, because I can only imagine you must be wearing like a spacesuit.
Unfortunately, it's not quite that bad. Although we have done a lot of work in astronaut genetics, and so I would love it if it was a spacesuit. I'd say it's a different subject for a different day. But you do have to. The PPE is covering the face, as well as multiple layers of gloves, as well as gowns. And we're still collecting patients. Actually, I have a person in the right now at the hospital. Chris Mazzari's from my lab, who's collecting samples and just doing oral swabs to work on a really rapid test we call LAMP, and that's in the paper we just put online with the patient data, which is a way where you can get a result basically within about 10 to 15 minutes of whether you're shedding the virus. So there's two kinds of testing. There's the serology and virology. You want to see, like, do you have antibodies to the virus? Did you have it before, or are you actively carrying the virus and shedding? And that's the virological testing. So we have a test that can detect the virus really quickly, and we're trying that out right now in patients. But even just in front of the patients and collecting them, actually patients have been, great, because I think 3 weeks ago, most of the patients were absolutely terrified because it was just the whole city felt like a movie that was an awful movie to be in. And now I think, the sun's coming out a bit more. I think most patients are, people know more now. So there's a time when it was just so new. Everyone was afraid. They didn't know what it would mean, who would die, who would get sick, who would get intubated, all these questions. But there's still a lot of fear in the eyes of the patients, but it's not as bad. If anything, the burnout is an issue for a lot of the clinical staff, because I've been working crazy for 6 weeks straight, and I think I've started to plan for a vacation in August that I'm presuming will be possible by then. I hope so. Yeah, my wife already bought tickets. Yeah.
Oh, well, I hope so too.
Yeah,
People have different debates about this, and you just alluded to this around burnout of the first responders and just people in general, just being fried from having so much fear. There's that level. And then there's the socioeconomic burden, right? So all of the people out of work, the different industries that are collapsing, like travel industry and other industries. And so I'm wondering, is there a way to phase this integration, kind of moving away from being at home and into further testing so that we can better gauge who can be out and actually keeping things running?
Yeah, and that's something that the donation to Cornell has helped a lot with. That we're trying to get as many methods out as possible that are high scalable methods for testing. So, I was even just thinking, I mentioned this briefly in the beginning, like thinking about people going back to school. You're going to want to test tens of thousands of people per week, or hundreds of thousands of people per week in a lot of municipalities, for peace of mind. I mean, I know I've had myself tested multiple times, because if you get too close to a patient, or there's a sample you think was contaminated, you're just worried. There's something really calming about knowing, okay, I've been tested, it looks like I'm negative. You feel like you can go back to your family, you can go home. So it's that fear is not going to go away. The virus is very contagious. It looks like it's sticking around. So we're working on everything from these PCR-based methods, which are called RT-PCR, which are kind of the current generally standard, but also LAMP is one method, called loop-mediated isothermal amplification. There's some other companies, I'm not involved with them, but just interesting, like Mammoth Biosciences and Sherlock, which make these little paper strips where you can actually do a quick detection at home. I think basically we're going to need to do three things, which is to roll out bigger testing in central labs, like at hospitals, big centers, begin to rolling out smaller places like pharmacies, pop-up kind of lab ideas. And then the third is to get home testing, so more people can be just in the comfort of their home, take a test, and make it cheap and make it effective. It's got to be cheap, because a lot of people have really bad insurance or no insurance. So if you say, hey, this test is only $50, for a lot of people, they might do it once, but they won't do it 3 or 4 times, because that's a lot of money to a lot of people. So I think, especially given the fact that, now at this point, 1 out of 5 people have no job, right? So $50 might not sound like a lot, but I think a lot of the tests have to come down to $2 or $3 a test to make it so people can keep themselves safe.
And how does it work for you, since you are, again, on the front lines and you're dealing with samples that have been infected? Are you testing just to that point, are you testing yourself on a daily basis, on a weekly basis? I mean, how often do you test to see whether or not you've actually been exposed?
More when it's only been every about week or two. Because I feel like we've been trying to conserve as many of the supplies as possible for people that we know need testing urgently. They're, for example, in the ICU. But now the supplies have stabilized. I think frontline workers should be tested, weekly or whenever they want to, basically. So I actually have a call right after I get off this line is with my brother, who's the mayor of Racine, Wisconsin, and we want to set up testing for all the frontline workers there. We want to do it for I'm from Wisconsin, hence all the Wisconsin references. But we want to make it so all the police and fire and healthcare workers, if they want to get tested once a week, it should just be free and they can get tested. So I'm having a call there to go over logistics. We're going to start to hopefully ramp this up Thursday this week and begin the first testing of people there.
Sounds good. Well, I want to be cognizant of your time. Again, you've been just incredibly busy, and we're so fortunate to be able to have you for this interview. Dr. Mason, can
Pleasure.
you leave us with any message of hope? You know, being that you are on the front lines, it feels rather hopeful in terms of things quieting down at the hospital front, people not having as much fear. But then I'm also hearing that we might be in for something even worse in the months to come. And so, what would you say for everybody out there? You know, is there a message of hope that we can bring forward to everybody in the way of being preventative and proactive with their health?
I think, definitely the people, it is simple things like, make sure you eat well and get sleep and try and think of things to do. If you're with families, do puzzles, do things, do some other games. If you're alone, Zoom with your friends or video conference with them, and don't forget about that we're a social species. And so don't lose that part of yourself. And also it will, even though it seems like, the world has never been more connected, so never a greater risk of having a pandemic, we've also never had the ability to track and monitor and respond to a virus as fast. So we already have, there's dozens of therapies being tried. There's also vaccine work being done on multiple fronts. We're able to track and exactly map where the virus is mutating, so we know almost everything there is to know about the virus at the genetic level, and then what it's doing to the patients and once it's in the body. So if you think of SARS, that was, the first SARS that came 17 years ago, none of those things I just mentioned were possible. So we just were blind to what the virus was, what it was doing, and where it was going. But now we see all of that. So I think from the epidemiology and the science and the medicine side, we have never had more tools or power and collaborative efforts. So I think that's really hopeful. And virus has been with us since as long as life has been around. So yeah, this is it's in some ways can be terrifying, but also this is part of life on this planet. So maybe on a different planet someday there'd be no viruses, but we're not there yet.
We're
So
not
this is
there yet, that's for sure.
just yeah, we just keep going. Yeah.
And it's like we are a microcosm of the planet. If we can just take care of our own microcosm, perhaps that will change the collective, the macrocosm of where
That's
we
right.
live.
That's right.
You know, because you really understand the exposome and the
Right.
condition that we're in just on the individual and also the planetary level. Dr.
Yeah.
Mason,
Yeah.
again, I just want to thank you so much. Again, I want to be cognizant of your time. Really appreciate you coming on. And I want to mention to all of our viewers that we have Giving Tuesday now. It's a global day of giving and unity. It's going to be taking place tomorrow, Tuesday, May 5th, Cinco de Mayo, as a response to the unprecedented need caused by COVID-19. If you're enjoying these talks, these expert interviews, and would like to get some information, we have our own position paper on the Resource Hub, which can be found at theana.org. So, and if you'd like to invest as part of our Giving Tuesday now, you'll also find that on the website, theana.org/invest. So thanks again, Dr. Mason. All the best to you. Stay healthy. We are incredibly grateful to you and your acts of service. And we really look forward to finding out what you find out. And so
Yeah.
keeping us apprised so that we will be better able to accommodate what happens potentially in the coming months. Thanks so much.
Sounds great. Thank you.